A single drop of ocean water contains 1 million living organisms, each one complete, each one alive, swimming, eating, reproducing, all invisible to your eyes. These are bacteria, the most successful life forms on Earth. They outnumber every other organism combined, outweigh all plants and animals together, and most are so tiny that hundreds could fit across a single human hair strand. The smallest known bacteria are called mycoplasma, about 2/10 of a micrometer wide. A micrometer is 1/1,000,000th of a meter. 5,000 of these tiny cells lined up would barely cross a grain of salt. If you enjoy these peaceful explorations of science, I would love for you to subscribe. Join this community, finding calm in curiosity. Leave a comment about where you are in the world. What time is it there? Do you think about the microscopic life around you? I love knowing we are scattered across time zones, all finding rest in small wonders together. This is life stripped to its core. No nucleus, no organs, no specialized compartments, just DNA floating in gel, ribosomes making proteins, a membrane holding everything together. The absolute minimum needed to be alive. Make a bacterium any smaller and something essential would not fit. The DNA would not have room. The ribosomes could not function. This is the limit. Life cannot be simpler and still work. Your cells are giants compared to bacteria. A typical human cell is 250 times larger than mycoplasma. If the bacterium were a marble, your cell would be a beach ball. But bacteria achieve with elegance what your cells achieve with complexity. Your cells contain dozens of structures: mitochondria for energy, endoplasmic reticulum for protein processing, Golgi apparatus for packaging, lysosomes for waste, all coordinated, all necessary. Bacteria skip this entirely. They make energy in their membrane. No mitochondria needed. They process proteins where they make them. No special compartments. Everything happens in one open space. Pure efficiency. This simplicity is not weakness. It is power. Bacteria reproduce faster than complex cells, survive in more environments, adapt to change in days instead of generations. They were here 3.5 billion years ago. They will likely outlast everything else. Your body hosts trillions of these organisms right now. On your skin, in your mouth, throughout your gut. More bacterial cells than your own cells. They are part of you, working with you, invisible partners in your existence. A speck of dust is a mountain to bacteria. Your skin is a landscape of ridges and valleys. They navigate this terrain their entire lives. Your body is their world, the only world they know. Water feels different at their scale, thick, viscous, like honey to you. Swimming requires rotating flagella, long tails that spin hundreds of times per second, propellers pushing through resistant liquid. Movement is hard work at microscopic dimensions. Temperature affects them instantly. No mass to buffer changes. No insulation to protect them. The environment's temperature becomes their temperature in milliseconds. They cannot regulate it, cannot resist it, just experience it directly. Yet bacteria thrive everywhere. In boiling springs, in frozen ice, in acid, in salt, in radiation, conditions that would destroy complex life. They adapt, survive, multiply. Their strength is their simplicity. They need almost nothing. A few molecules, some energy. That is enough. While complex organisms require stable conditions and abundant resources, bacteria make do with whatever exists. Right now, bacteria surround you completely, in every breath, on every surface, floating in air, living in water, coating soil. Most are harmless, many are helpful. All are busy with their simple chemistry, converting molecules, storing energy, copying DNA, being alive in the most basic way possible. And there is peace in understanding this. Life, at its foundation, is chemistry. Molecules interacting according to physical laws. No mystery, no magic, just elegant simplicity that has worked for billions of years. You share this planet with more bacteria than stars in the galaxy. They are everywhere, always, working quietly, cycling nutrients, producing oxygen, breaking down waste, making life possible for everything else. Tonight, as you rest, bacteria rest too. Some forming spores, entering dormancy, waiting for better conditions. Others dividing, doubling, creating the next generation. All following patterns older than mountains, simpler than thought. Pure existence at the foundation of all life. A speck of dust is a mountain to bacteria. Your skin is a landscape of ridges and valleys. They navigate this terrain their entire lives. Your body is their world, the only world they know. Water feels different at their scale. Thick, viscous, like honey to you. Swimming requires rotating flagella, long tails that spin hundreds of times per second, propellers pushing through resistant liquid. Movement is hard work at microscopic dimensions. Temperature affects them instantly. No mass to buffer changes, no insulation to protect them. The environment's temperature becomes their temperature in milliseconds. They cannot regulate it, cannot resist it, just experience it directly. Yet bacteria thrive everywhere. In boiling springs, in frozen ice, in acid, in salt, in radiation, conditions that would destroy complex life. They adapt, survive, multiply. Their strength is their simplicity. They need almost nothing. A few molecules, some energy. That is enough. While complex organisms require stable conditions and abundant resources, bacteria make do with whatever exists. Right now, bacteria surround you completely, in every breath, on every surface, floating in air, living in water, coating soil. Most are harmless, many are helpful. All are busy with their simple chemistry, converting molecules, storing energy, copying DNA, being alive in the most basic way possible. And there is peace in understanding this. Life at its foundation is chemistry, molecules interacting according to physical laws. No mystery, no magic, just elegant simplicity that has worked for billions of years. You share this planet with more bacteria than stars in the galaxy. They are everywhere, always, working quietly, cycling nutrients, producing oxygen, breaking down waste, making life possible for everything else. Tonight, as you rest, bacteria rest too. Some forming spores, entering dormancy, waiting for better conditions. Others dividing, doubling, creating the next generation. All following patterns older than mountains, simpler than thought, pure existence at the foundation of all life. For most of human history, no one knew these organisms existed. The invisible world stayed invisible, until one man in Holland started grinding lenses. Antony van Leeuwenhoek, a fabric merchant in Delft. The year was 1674. He had no formal education, no scientific training, just curiosity and incredibly skilled hands. He ground tiny lenses, smaller than anyone else made. Each lens only a few millimeters across, like glass beads, but perfectly shaped. These lenses could magnify nearly 300 times, far more powerful than any microscope of his era. Van Leeuwenhoek mounted these lenses in small brass holders, made simple microscopes. Just a lens and a holder, a pin to hold specimens. Nothing fancy, but effective. He looked at everything: pond water, rainwater, his own saliva, scrapings from his teeth. The world became his laboratory, every substance a potential discovery. One day, he examined a drop of water, saw movement, tiny creatures swimming, smaller than anything he had ever imagined. He called them animalcules, little animals. He wrote letters to the Royal Society in London, described what he saw, drew pictures. Thousands of these creatures in a single drop, moving, spinning, alive. The scientists did not believe him at first. Invisible animals? Impossible. Too small to exist. He must be mistaken. Seeing artifacts. Dirt on his lens, perhaps. But Van Leeuwenhoek was meticulous. He repeated his observations, made better drawings, sent more letters. Eventually, other scientists built microscopes, looked for themselves, confirmed his discoveries. The invisible world became visible. Life existed at scales no one had conceived. Everywhere Van Leeuwenhoek looked, he found these animalcules. In every drop of water, on every surface, the world was teeming with invisible life. He discovered bacteria, protozoa, blood cells, sperm cells, opened entire realms of biology, all with lenses he ground by hand, all through careful observation and patience. His tooth scrapings were particularly shocking. He found countless tiny organisms living in the plaque between his teeth, wrote that there were more animalcules in his mouth than people in all of Holland. He was right. Your mouth contains billions of bacteria, more bacterial cells than human beings alive. Van Leeuwenhoek cleaned his teeth with salt, vinegar, hot coffee, checked again under his microscope. Many animalcules died from these treatments, but some survived. Nothing killed them all. They were too diverse, too adaptable. He kept observing for 50 years, sending letters, making discoveries. Never stopped being amazed by what he saw. The invisible world never became ordinary to him. Each observation brought fresh wonder. Other scientists built on his work. Better microscopes, more powerful magnification. Understanding grew slowly. These animalcules were not little animals. They were something different, simpler, more basic. By the 1800s, scientists realized these organisms were single cells, complete life forms in one cell, not tiny versions of larger animals, fundamentally different, a separate category of life. They named them bacteria, from Greek word meaning little rod, because many bacteria are rod-shaped, simple names for simple organisms. The discovery changed medicine, changed science, changed how humans understood the world. Disease made sense suddenly. Infection had a cause. These invisible organisms invading the body, multiplying, causing harm, but also helping. Bacteria in the gut aiding digestion. Bacteria in soil helping plants grow. Bacteria everywhere doing essential work. Life depending on these invisible helpers. Van Leeuwenhoek never knew the full importance of his discovery. He just kept looking, kept observing, kept writing letters about what he saw, curiosity driving him forward. He died in 1723, 90 years old, spent his last decades still grinding lenses, still looking through his microscopes, still finding wonders in drops of water. His microscopes were lost, his techniques forgotten. No one else could grind lenses quite like he did. But his discovery remained. The invisible world he revealed. Now we have electron microscopes, can see viruses smaller than bacteria, can see individual atoms. Technology Van Leeuwenhoek could not imagine. But it started with him, with hand-ground lenses, with looking at pond water, with the simple question of what exists beyond what eyes can see. Tonight, those same organisms swim in water everywhere, live on every surface, exist in numbers beyond counting, still invisible to your eyes, but real, alive, discovered by one curious man with homemade lenses. The world within the world, revealed. Van Leeuwenhoek saw bacteria from the outside, swimming shapes, moving dots. But what is inside? What makes them work? A bacterium is a single cell, everything needed for life packed into one microscopic package. No separate parts, no divisions, just one compartment holding everything. The outer boundary is a membrane. Think of it as a bag, made of fats and proteins. This membrane holds the cell together, keeps the inside in and the outside out, controls what enters and leaves. Some bacteria have a cell wall outside the membrane, made of peptidoglycan, a mesh of sugars and proteins. This wall is rigid, gives the bacterium shape, protects it from bursting when water rushes in. Inside the membrane is the cytoplasm, a gel-like substance. Water mixed with proteins and salts. Everything floats in this gel, the cell's workshop, where chemistry happens. The DNA floats free in the cytoplasm, not packaged in a nucleus. No membrane separating it from the rest of the cell. Just a long loop of DNA drifting in the gel. This loop contains all the instructions for being a bacterium. How to make proteins, how to reproduce, how to survive. The DNA in a bacterium is surprisingly long. If you stretched it out, it would be 1,000 times longer than the cell itself. It stays compact by coiling, twisting, folding back on itself, like a spring compressed tight. Scattered throughout the cytoplasm are ribosomes, tiny machines made of RNA and protein. These read the DNA instructions, build proteins according to those instructions. Each ribosome is a factory, taking raw materials and assembling them into useful molecules. A single bacterium might have 20,000 ribosomes, all working constantly, making enzymes, structural proteins, everything the cell needs. The ribosome is essential. Without it, the DNA instructions are useless, just information with no way to act. That is nearly everything. Membrane, cell wall, DNA, ribosomes, cytoplasm. This minimal set creates a living organism. Nothing else is required. No mitochondria. Bacteria make energy directly in their membrane. No need for a separate organelle. The membrane itself does the work. No endoplasmic reticulum. No Golgi apparatus. No lysosomes. None of the structures your cells depend on. Bacteria function without them. Proof that life does not need complexity. Some bacteria have flagella, long whip-like tails for swimming. These extend from the cell, powered by a motor embedded in the membrane, the simplest rotating machine in nature. But even flagella are optional. Many bacteria lack them, stay still, live their whole lives in one spot. Some have pili, hair-like projections, used to stick to surfaces, to exchange DNA with other bacteria. But again, optional, not required for life. The truly essential parts fit on a short list. Membrane, DNA, ribosomes. Everything else is specialization, adaptation, useful but not necessary. This minimalism is beautiful. Life stripped to its core. No decoration, no excess, pure function. Each component doing exactly what is needed. Nothing wasted. Your cells are the opposite. Compartmentalized, specialized. Thousands of different proteins. Intricate structures. Multiple membranes dividing the cell into sections. Complexity creating capability. But bacteria prove complexity is not required. They reproduce faster than your cells, survive in more environments, exist in greater numbers. Simplicity works. The bacterium is a reminder that life is chemistry, molecules interacting, information encoded in DNA, proteins carrying out tasks, membranes separating inside from outside. Nothing mystical, nothing beyond physics and chemistry. And yet, it is alive. It grows, responds to its environment, reproduces, maintains itself against entropy, creates order from disorder, all with the bare minimum of parts. This is what makes bacteria fascinating. They are life reduced to essentials. The answer to the question, what is the least you need to be alive? A membrane, DNA, ribosomes, cytoplasm. That is enough. Everything else is optional. Evolution has tested this design for 3.5 billion years. It works. Simple, elegant, sufficient. Right now, trillions of these minimal cells surround you, each one functioning with just these basic parts. Living proof that complexity is not necessary, that life can be simple and still complete, still whole, still alive in every meaningful sense. Those simple cells live at a scale difficult to imagine. Let me help you picture it. Take a standard pin, the kind you use for sewing. Look at the head, that small round tip. Thousands of bacteria could live comfortably on that surface, not cramped, comfortably, with room to move. If bacteria were visible to your eyes, a pinhead would look like a vast plain. Room for cities, for territories, for entire populations to exist without crowding. One type of bacteria, called Staphylococcus, is about 1 micrometer across, a sphere 1/1,000,000th of a meter wide. The head of a pin is about 2 millimeters, 2,000 micrometers. You could line up 2,000 bacteria across the pinhead in one direction, 2,000 in the other direction. That is 4 million bacteria on the surface of a pinhead. But bacteria are three-dimensional. They stack. A pinhead could hold millions more in layers. A small mountain of bacteria, all living in the space you barely notice. From their perspective, the pinhead is enormous. The texture of the metal is a landscape. Tiny imperfections become canyons. Scratch marks become valleys. What looks smooth to you is rough terrain to them. A grain of salt would be a massive boulder. Table salt crystals are about 300 micrometers on each side. To a 1 micrometer bacterium, this crystal is 300 times its own width. Imagine a cube 300 times wider than you are tall. That is how a grain of salt appears to a bacterium. A human hair is a vast column, about 70 micrometers wide, 70 times the width of our bacterium, like a tree trunk to you, but wider, more massive. Bacteria live on human hairs, grip the ridged surface, navigate around it. The hair is their environment. Water droplets are oceans. A droplet from a mist might be 50 micrometers across. To bacteria, this is a sphere of water large enough to contain their entire world. They swim in it, find food in it, reproduce in it. When the droplet evaporates, their ocean disappears. Dust particles are mountains. Most dust is between 1 and 100 micrometers. The larger pieces dwarf bacteria. These particles float through air, land on surfaces, carry bacteria with them. The dust becomes a vehicle, transportation to new locations. Distance means something different at this scale. A bacterium might travel a few millimeters in its lifetime. This seems like nothing to you, but to the bacterium, it is thousands of body lengths, like you walking hundreds of miles. Speed is relative too. Some bacteria swim at 50 micrometers per second. This sounds slow, but relative to their size, it is fast. 50 times their body length per second. equivalent to you swimming 50 meters per second, faster than any human can move in water. Time perception might differ as well. Chemical reactions happen at the same speed regardless of scale. But a bacterium lives its whole life cycle in hours. Rapid existence, fast reproduction. What feels like a lifetime to the bacterium is less than a day to you. The forces they experience are different. Gravity barely affects them. They are too small, too light. Surface tension dominates instead. Water molecules stick together more strongly than gravity pulls the bacterium down. This makes water thick, almost solid, like living in honey. Air currents are violent storms. A gentle breeze to you is hurricane force to them. Air molecules buffet them constantly. Random motion called Brownian motion. They are pushed and jostled by invisible collisions with air and water molecules. Temperature changes hit them instantly. No thermal mass, no insulation. The environment's temperature becomes their temperature in milliseconds. They cannot regulate it, cannot buffer against it, just experience it directly. This is the world bacteria inhabit, where pinheads are vast, where salt grains are boulders, where hair is landscape, where water is thick, where air is violent, a parallel world, existing at a scale we cannot directly perceive, overlapping with ours, but fundamentally different. Right now, bacteria navigate this microscopic world on every surface around you, living at their scale, experiencing their reality, completely unaware of your existence. You are too large, too far beyond their perception, just as their world is beyond yours, without help from microscopes. Two realities, same space, different scales, both completely real, both fully alive. In that microscopic world, bacteria face a problem you solve with every breath. They need oxygen, but they have no lungs, no gills, no breathing apparatus at all. They use diffusion, the simplest possible solution. Oxygen dissolves in water, in the moisture surrounding bacteria. Oxygen molecules bounce randomly through this water. Eventually, some molecules hit the bacterial membrane, pass through, enter the cell. Inside the cell, oxygen gets used immediately, consumed by chemical reactions. This creates a concentration difference. More oxygen outside the cell than inside. This difference matters. Molecules naturally move from high concentration to low concentration, like a crowd spreading from a packed room into an empty hallway. No force needed, just random motion creating flow. Oxygen outside the bacterium is abundant. Oxygen inside gets consumed constantly. This concentration difference drives oxygen inward, through the membrane, into the cell, automatically, continuously. The bacterium does nothing active, does not pump, does not breathe, just exists. Oxygen flows in because of physics, because of concentration differences, because of random molecular motion. This works because bacteria are small, extremely small. Size makes all the difference. Oxygen entering at the membrane needs to reach the center of the cell. In a large organism, this distance is huge. Oxygen would take too long to diffuse. The center would suffocate before oxygen arrived. But in a bacterium, the center is only half a micrometer away. Oxygen molecules reach it in milliseconds. Diffusion is fast enough. No pumping needed. This is why bacteria can be passive breathers. Their smallness saves them, makes diffusion sufficient. There is a mathematical relationship, surface area versus volume. As organisms get larger, volume grows faster than surface area. A bacterium doubled in size, has 4 times the surface area, but 8 times the volume. More volume means more cells needing oxygen, but less surface area per volume means less membrane for oxygen to cross. Eventually, diffusion cannot keep up. The organism needs active breathing, pumping blood, circulating oxygen. Bacteria never reach this point. They stay small enough that their surface area is large relative to their volume. Plenty of membrane for oxygen exchange, plenty of proximity to the center. Some bacteria need very little oxygen. They make energy through fermentation instead of respiration. These bacteria can survive in environments with almost no oxygen. Deep in soil, in sealed containers, in your intestines where oxygen is scarce. Others need oxygen constantly. These aerobic bacteria live in oxygen-rich environments, near water surfaces, in well-aerated soil, on your skin exposed to air. The same diffusion principle works in reverse for waste. Carbon dioxide builds up inside the cell, creates high concentration there, low concentration outside. Carbon dioxide diffuses out, through the membrane, into the environment. No exhalation needed, no active removal, just concentration differences driving flow. In with oxygen, out with carbon dioxide. Passive exchange. Temperature affects diffusion speed. Warmer water means faster molecular motion. Oxygen diffuses faster. Bacteria in warm environments get oxygen more quickly. Cold slows everything, makes diffusion sluggish. Bacteria in cold water must manage with slower oxygen delivery. The membrane matters too. It must be permeable to oxygen. Some molecules cannot pass through membranes easily, but oxygen is small, non-polar, slips through the fatty membrane without difficulty, like dissolving into oil. This elegant system has worked for billions of years. Bacteria invented it first. Were breathing through diffusion before complex life existed, before lungs, before gills, before blood. Your cells use diffusion too, after your lungs deliver oxygen to your blood, after your blood delivers it to tissues. Individual cells absorb oxygen through diffusion. Same principle, same physics. But you cannot rely on diffusion alone. You are too large. Your cells are too far from outside air. You need the whole system. Lungs, heart, blood, all working to deliver oxygen where diffusion can take over. Bacteria skip all that. Stay small. Let physics do the work. Simple, efficient, sufficient. Right now, bacteria around you are breathing through diffusion. Oxygen flowing in, carbon dioxide flowing out. No effort, no machinery, just molecules following concentration gradients. Life at its simplest, chemistry in action, working perfectly at the smallest scale. Bacteria absorb oxygen through their membrane. They absorb food the same way, but food is more complicated than oxygen. Oxygen is a small molecule, slips through membranes easily. Food is different. Proteins, sugars, fats. These molecules are large, too large to pass through the membrane directly. Bacteria solve this with chemistry. They digest food outside their bodies. Picture a bacterium on a piece of bread. The bread is mostly starch, long chains of sugar molecules linked together. These chains are enormous compared to the bacterium. Cannot enter the cell. The bacterium releases enzymes, special proteins that break chemical bonds. These enzymes float out through the membrane, spread into the surrounding bread, start cutting the starch chains. Snip. A long chain becomes two shorter chains. Snip again. Shorter still. Eventually, the enzyme cuts the chain down to individual sugar molecules. Glucose. Small enough to cross the membrane. This is digestion, the same process that happens in your stomach, but happening outside the cell. In the environment. The bacterium's digestive system is the world around it. The sugar molecules diffuse back toward the bacterium. Concentration is high near the cell, where enzymes are working. Low inside the cell, where sugar gets consumed. The gradient pulls sugar inward. Transport proteins in the membrane help. These proteins grab sugar molecules, pull them through the membrane, deliver them inside. Active transport. The bacterium does spend energy here, but only for the final step. Different enzymes break down different foods. Proteases cut proteins into amino acids. Lipases break fats into fatty acids. Each enzyme specialized for one type of chemical bond. Bacteria release the enzymes they need for whatever food is available. Sense what is in the environment. Respond by making appropriate enzymes. Flexible, adaptable. Some bacteria release enzymes constantly, digest everything around them. Others are more selective. Only release enzymes when specific foods are detected. This saves energy. No point making enzymes for food that is not there. The digestion creates competition. Multiple bacteria might release enzymes into the same food source, all breaking it down, all absorbing the results. Whoever is closest, whoever has the most efficient enzymes, whoever absorbs fastest, they get the most nutrition. This external digestion changes the environment. A piece of food becomes a cloud of smaller molecules. Sugar, amino acids, fatty acids, all floating in solution, all available to any bacterium nearby. Bacteria do not hunt, do not gather, do not move food to their mouths. They live in food, or food lives around them. They bathe in nutrients, absorb what they need directly. Some nutrients are already small enough. Dissolved minerals, simple sugars in fruit. These pass through the membrane without pre-digestion. Straight in, ready to use. Others require processing. Cellulose from plant cell walls. Extremely tough. Long chains of glucose bound in ways most organisms cannot break. But some bacteria have enzymes for this. Can digest cellulose. Live on plant material other organisms cannot use. This ability is crucial. Cows cannot digest grass. They rely on bacteria in their stomachs to do it. The bacteria release cellulase enzymes, break down cellulose, release sugars. The cow absorbs these sugars from the bacteria's work. Your intestines work similarly. You cannot digest certain plant fibers. Bacteria in your gut can. They release enzymes, break down fibers, produce vitamins, short-chain fatty acids. You absorb these products, benefit from bacterial digestion. Bacteria can eat almost anything organic. Wood, fabric, dead leaves, animal tissue. Wherever there are organic molecules, some bacterium has enzymes to digest them. Some even eat inorganic materials. Sulfur bacteria oxidize hydrogen sulfide. Iron bacteria process iron compounds. These bacteria get energy from chemistry we consider purely mineral. Life finding a way in unexpected places. The membrane is both barrier and doorway, keeps the cell intact, but allows selective passage. Small molecules in, waste products out, enzymes out to digest, nutrients in to fuel life. No mouth needed, no stomach, no intestines, just membrane, enzymes, diffusion, chemical processes happening in open water, in soil, on surfaces, wherever bacteria live. Right now, bacteria are digesting food all around you, releasing enzymes into their environments, breaking down molecules, absorbing nutrients, feeding without eating. Life as pure chemistry. Simple, effective, enough. Bacteria absorb food through their membrane. This only works because they live surrounded by water. Water is their entire world. Even bacteria on dry surfaces live in water. A thin film of moisture covers everything, invisible to you. But to bacteria, it is an ocean. They swim in this film, never truly dry. Water at bacterial scale behaves strangely, not like the water you experience. The physics change when you are that small. To you, water flows easily, pours, splashes, moves freely. To a bacterium, water is thick, viscous, like honey, or thin syrup. Moving through it requires effort. This happens because of something called the Reynolds number. It compares inertial forces to viscous forces. At large scales, inertia dominates. You can glide through water after a single push. Momentum carries you forward. At bacterial scale, viscosity dominates. No momentum, no gliding. Stop swimming and you stop moving instantly. The water grabs you, holds you in place. Imagine swimming in honey. Push forward, move a little, stop pushing, freeze immediately, no coasting. This is bacterial swimming. To move, bacteria use flagella, long whip-like structures extending from the cell. Each flagellum is a thin filament, about 20 nanometers wide, much thinner than the bacterium itself. The flagellum connects to a motor at its base. This motor is embedded in the cell membrane, made of proteins arranged in rings. The motor spins. The flagellum rotates with it. This is the only true rotary motor in biology. Everything else moves by contraction or bending. Muscles pull, cilia wave. But the bacterial flagellum rotates completely, spins like a propeller, 360-degree rotation. The motor spins fast, 100 to 300 revolutions per second, faster than your eye could follow if you could see it. The rotating flagellum pushes against the thick water, propels the bacterium forward. A bacterium with one flagellum at its back end swims in a straight line. The flagellum spins counterclockwise, acts as a propeller, pushes the bacterium forward. Simple, efficient. Some bacteria have multiple flagella, all over their surface. When these flagella spin the same direction, they bundle together, form a single rotating mass, push the bacterium forward in a straight line. When the flagella reverse direction, the bundle flies apart. Each flagellum goes its own way. The bacterium tumbles, spins randomly. This is how bacteria turn. Swim straight, tumble, swim in a new direction, tumble again. This creates a random walk. The bacterium explores its environment without knowing where it is going. If it swims toward food, chemical sensors detect increasing concentration. The bacterium tumbles less often, continues swimming straight more. This biases the random walk toward food. Not all bacteria can swim. Many have no flagella. These bacteria stay where they are, drift with currents, live their entire lives in one spot, still successful, still alive. Movement is optional. For swimming bacteria, the thick water creates challenges. They can move, but not far. A bacterium might swim a few millimeters in its lifetime. To you, this is nothing. To the bacterium, it is thousands of body lengths. Significant distance at their scale. The energy cost is high. Running the flagella motor takes resources. About 2% of the bacterium's total energy goes to swimming. This is why many bacteria do not bother. Save that energy for reproduction instead. Swimming also requires precision engineering. The flagellar motor has dozens of parts. All must fit perfectly, work together. One protein out of place, and the motor fails. The bacterium cannot swim, but when it works beautifully. A molecular machine, rotating at hundreds of times per second, pushing through viscous water, giving a single cell mobility. the ability to seek food, escape danger, find optimal conditions. Water is both medium and challenge. Bacteria need it to survive, cannot live without moisture, but moving through it requires special adaptations. Molecular motors, rotating flagella, energy expenditure. Right now, bacteria swim in water all around you, in the moisture on your skin, in the film on surfaces, in actual water if it is nearby. Each one navigating a thick, viscous world, swimming through what feels like honey, moving because tiny motors spin, life in motion at the smallest scale. Bacteria swim through water, but their most remarkable ability is not movement. It is reproduction. Pure, simple, fast. Bacteria reproduce by dividing in two. One cell becomes two. Two become four. Four become eight. Exponential growth. The process is called binary fission. Binary means two. Fission means splitting. Two-way splitting. The name describes exactly what happens. It starts with DNA. The bacterium copies its entire genome. Every gene. Every instruction. Now, the cell contains two complete sets of DNA instead of one. These DNA copies attach to different points on the cell membrane. As the membrane grows, it pulls the DNA copies apart, moves them toward opposite ends of the cell. Physical separation through growth. The cell elongates, gets longer, stretches. A new membrane forms down the middle, begins as a small fold, grows inward from all sides, eventually meets in the center, seals, divides the cell into two compartments. Each compartment contains one copy of the DNA, half the ribosomes, half the cytoplasm, everything distributed roughly equally. Both halves are complete cells, both alive, both capable of repeating the process. The original cell no longer exists. It did not die, it became two cells. Neither is the parent, neither is the child. Both are continuations. Both are the original cell transformed. This is fundamentally different from how you reproduce. You create offspring separate from yourself. You remain. Your children are new individuals. Bacteria do not work this way. They transform, persist through division, are immortal in a sense. Every bacterium alive today is the same cell that existed billions of years ago, just divided trillions of times. The speed is remarkable. Some bacteria divide every 20 minutes. One cell, wait 20 minutes. Two cells, wait 20 more. Four cells. In 1 hour, one bacterium becomes 8. In 24 hours, one bacterium becomes millions. If nothing stops the growth, if food and space are unlimited, the mathematics is explosive. Of course, nothing is unlimited. Food runs out, space fills, waste accumulates, growth slows, stops, populations crash. This is the pattern. Boom, bust, cycle repeating. But the potential exists, the machinery for rapid reproduction. Bacteria can exploit resources quickly, colonize new environments, respond to opportunities faster than complex organisms. No courtship needed, no mate finding, no pregnancy, no childhood, just growth, division, two cells where one existed, both ready to repeat immediately. Mutations happen during copying. DNA replication is not perfect. Errors occur. Most errors are harmful, kill the cell, or slow its growth. But occasionally, an error creates advantage. Maybe an enzyme becomes more efficient. Maybe a protein provides resistance to antibiotics. That mutant cell divides, passes the advantage to its descendants. Rapid reproduction means rapid evolution. This is why antibiotic resistance spreads so fast. A resistant bacterium can create millions of resistant descendants in days. Selection favors the resistant cells. Soon, the population is mostly resistant. Evolution in real time. The division process requires coordination. DNA must copy completely before division. If the cell divides too soon, one daughter gets incomplete DNA, dies. If the cell waits too long, resources are wasted. Timing matters. Proteins regulate the process. Check that DNA is ready. Verify the cell is large enough. Confirm conditions are suitable. Only then allow division to proceed. Quality control at the molecular level. Some bacteria synchronize their division. Entire populations divide at the same time. This creates population waves, pulses of growth. The mechanism is not fully understood, but the coordination is real, observed in laboratories. Not all bacterial reproduction is division. Some bacteria exchange DNA, take in DNA from the environment. This is genetic mixing without reproduction, acquiring traits from other bacteria, sharing adaptations, horizontal gene transfer. But division remains the primary method, the core reproductive strategy. Simple splitting, no complexity needed, no elaborate mechanisms, just growth, DNA replication, membrane formation, separation. Right now, bacteria around you are dividing, populations doubling, millions becoming billions, all through simple cell division. No parents, no offspring, just transformation, continuation. The simplest reproduction, the fastest, the most efficient. Life multiplying through elegant simplicity. Bacteria divide rapidly, create vast populations, and they do this everywhere. Literally everywhere on Earth, in soil. Every handful contains billions of bacteria. They live between soil particles, on root surfaces, in tiny water pockets, breaking down organic matter, recycling nutrients, making soil alive, in water, oceans, lakes, rivers, puddles. Bacteria fill every drop. 1 million per milliliter in seawater, more in freshwater, more still in stagnant pools. The water is thick with invisible life. In air, every breath you take contains bacteria. They float on dust particles, ride water droplets, drift on wind currents. Fewer than in water or soil, but present, always present. On your skin, your entire surface is colonized. Bacteria live in the oils, in the moisture, between skin cells, millions per square centimeter. Different species in different areas. Oily skin hosts different bacteria than dry skin. In your mouth, even more bacteria. Billions. They coat your teeth, line your cheeks, cover your tongue, form biofilms, living communities stuck to surfaces. These bacteria are mostly harmless. Many are helpful. In your gut, the largest bacterial population in your body. Trillions of cells, hundreds of species. They outnumber your own cells. Fill your intestines. Aid digestion. Produce vitamins. Train your immune system. In extreme environments, places you would call uninhabitable, bacteria thrive there too. Hot springs above the boiling point. Bacteria live in them, survive, reproduce. Heat does not kill them. Deep ocean vents. Water at 400 degrees Celsius. Under crushing pressure. Bacteria live there. Use chemicals from the vents for energy. Build ecosystems in absolute darkness. Antarctic ice, frozen solid. Bacteria survive inside. Dormant but alive. Waiting for brief summer melts. Active for days. Then frozen again for years. Deep underground. Miles below the surface. In cracks in bedrock. Bacteria live there. No light. No oxygen. No organic matter. They eat rock. Process minerals. Generate energy from chemistry alone. In acid lakes. Water acidic enough to dissolve metal. pH near 0. Bacteria thrive. Their membranes adapted. Their proteins resistant. Living in acid that would destroy most life. In salt lakes. Water so salty it crystallizes. Bacteria live there. manage the salt concentration inside their cells, maintain balance, function normally in conditions that would kill other organisms, in radioactive waste, exposed to radiation that shreds DNA. Bacteria survive, repair their DNA constantly, tolerate doses thousands of times higher than would kill you. This adaptability is extraordinary. No environment too harsh, too hot, too cold, too acidic, too salty, too radioactive. Somewhere, some bacterium can live there. The key is diversity. Bacteria have had billions of years to evolve, to try every possible adaptation, to colonize every possible niche. Some lineages succeeded in extreme places, became extremophiles, but bacteria also live in normal places, on leaves, in clouds, on building surfaces, in carpets, in refrigerators, in shower heads. Wherever moisture exists, wherever organic molecules exist, bacteria find a way. They form the base of most ecosystems, primary producers, breaking down waste, recycling elements. Other organisms depend on them. Remove bacteria, and most ecosystems collapse, even inside other organisms. Bacteria live in insects, in plants, in fungi. Symbiotic relationships, mutual benefit. The bacteria gain shelter and food. The host gains nutrients or protection. Partnerships at the cellular level. Some bacteria live in clouds, high in the atmosphere, using water droplets as homes, feeding on organic molecules in air, influencing cloud formation, affecting weather in small ways. Some bacteria live in rocks, boring into stone, creating tiny chambers, living inside solid rock for generations, barely active, conserving energy, waiting. The total bacterial biomass on Earth is staggering, more than all plants combined, more than all animals. They are invisible, ignored, but they outweigh everything else. Right now, bacteria surround you, above, below, beside, on you, in every direction, every space filled, every surface colonized. Life at the smallest scale existing everywhere simultaneously. The most successful organisms on Earth, living where nothing else can, thriving in conditions we consider impossible, everywhere and always. Bacteria live everywhere, but some live in places that seem impossible. These are extremophiles, lovers of extremes. Thermophiles love heat. They live in hot springs, geysers, volcanic vents. Water above the boiling point does not kill them. It is their preferred temperature. The hottest known bacterium survives at 122 degrees Celsius, 252 degrees Fahrenheit. Water only stays liquid because of pressure. At normal pressure, this temperature creates steam instantly. How do they survive? Special proteins. Normal proteins unfold at high heat, lose their shape, stop working. Thermophile proteins are stabilized. Extra chemical bonds, different structures. They stay folded even when hot. Their membranes are different too. Lipids that remain stable at high temperature, do not melt or fall apart, keep the cell intact even in boiling water. Psychrophiles love cold. They live in Arctic oceans, Antarctic ice, permafrost. They grow best near freezing. Some can reproduce at -12 degrees Celsius, below the freezing point of pure water. They make antifreeze proteins. These prevent ice crystals from forming inside the cell. Ice would puncture membranes, destroy structures. The antifreeze keeps internal water liquid, even in freezing conditions. Their enzymes work when cold. Most enzymes become sluggish at low temperatures. Slow down, almost stop. Psychrophile enzymes stay active, flexible, functional even when frozen. Acidophiles love acid. They live in acid mine drainage, volcanic sulfur pools, places with pH near 0, acid strong enough to burn skin. Their membranes resist acid, do not dissolve. The inside of their cell is neutral, normal pH, but they pump protons out constantly, maintain the difference, survive in acid by keeping it outside. They use the pH difference for energy. Let protons flow back in through special proteins. This flow generates power. Like a hydroelectric dam, the acid becomes their energy source. Alkaliphiles are the opposite. They love bases, live in soda lakes, places with pH above 10, as alkaline as bleach. They face the same challenge backwards. Keep their insides neutral while outsides are basic. Pump ions to maintain balance. Use the gradient for energy. Same strategy, different extreme. Halophiles love salt. They live in salt lakes, salt evaporation ponds, the Dead Sea. Water so salty it forms crystals. They accumulate salt inside their cells. Match the outside concentration. This prevents water from leaving the cell. Normal cells would shrivel in salt water. Water flows out to balance concentration. Halophiles avoid this by becoming salty inside. All their proteins are adapted, work in high salt, require salt to function properly. Take a halophile and put it in fresh water. It dies. Too little salt, its proteins stop working. Radio-resistant bacteria survive extreme radiation. Some can tolerate doses 5,000 times higher than would kill you. Their DNA breaks constantly. Radiation shreds it, but they repair it just as fast. Multiple copies of their genome. If one breaks, they copy from another. Fix the damage. Continue living. Special proteins protect their other molecules. Soak up radiation damage. Keep essential structures intact. The cell sacrifices these proteins, makes more, keeps functioning. Barophiles love pressure. They live in deep ocean trenches, miles underwater. Pressure that would crush most organisms. Their proteins require pressure. They fold differently under compression. Use pressure to maintain shape. Take them to the surface and they fall apart. Too little pressure. Their proteins unfold. These extremophiles show life's limits, or lack of limits. Every extreme we test. Some bacterium survives, thrives even. This matters for astrobiology. If bacteria survive Earth's extremes, they could survive elsewhere. Mars, Europa, Enceladus. Wherever water and energy exist, life might exist too, in forms similar to Earth's extremophiles. The extremes also teach us basic biology, show what is essential, what can change, how flexible life can be. Push organisms to limits and they reveal what matters, what can be adapted. Right now, extremophiles live in places you cannot. Places that would kill you instantly. They are there, thriving, reproducing, perfectly comfortable in conditions we call extreme. Life adapting to every possible environment, finding ways to survive anywhere. These extremophiles survive because bacteria are ancient. They have had time to adapt, to evolve, to colonize every environment. 3.5 billion years of time. Bacteria were the first life on Earth. Not plants, not animals. Bacteria. They appeared when Earth was young, hostile, nothing like today. The earliest fossils are stromatolites, layered structures made by bacterial mats. These fossils are 3.5 billion years old. Bacteria living, building, creating stone through their metabolism. Modern stromatolites still exist, in a few places. Shark Bay in Australia, some hypersaline lakes, living fossils. Bacteria building the same structures their ancestors built billions of years ago. For 2 billion years, bacteria were the only life. Nothing else existed, just bacteria. In oceans, on rocks, forming mats, releasing chemicals, slowly changing the planet. The biggest change was oxygen. Early Earth had no oxygen in the atmosphere. Oxygen was toxic, reactive, destructive. The atmosphere was mostly nitrogen and carbon dioxide. Then, cyanobacteria evolved. These bacteria could photosynthesize, use sunlight to make food. A byproduct of their photosynthesis was oxygen. At first, the oxygen dissolved in ocean water, reacted with iron, created rust, fell to the ocean floor, formed iron deposits, banded iron formations, stripes of iron oxide in ancient rocks, evidence of bacterial oxygen. Eventually, all the iron was oxidized. Oxygen began accumulating in the atmosphere. This took millions of years. Slowly, the percentage increased. 1%, 2%, eventually 21%, the level today. This was catastrophic for most bacteria. Oxygen poisoned them, killed them, drove them to extinction or into oxygen-free environments. The Great Oxidation Event, mass extinction caused by bacterial metabolism. But some bacteria adapted, developed enzymes to handle oxygen, eventually learned to use oxygen for energy. Aerobic respiration, more efficient than anything before. These bacteria thrived in the new oxygen-rich world. Your cells use aerobic respiration, inherited from bacteria. Your mitochondria are descendants of ancient bacteria. They entered other cells, formed partnerships, became organelles, but they still carry bacterial DNA, still have double membranes, still show their bacterial ancestry. Chloroplasts are the same, descendants of cyanobacteria, the bacteria that made oxygen. They entered plant cells, became organelles. Now, they make food for plants through photosynthesis, the same process their ancestors invented billions of years ago. Every complex cell on Earth, every plant, every animal, every fungus, all descended from simpler cells that absorbed bacteria, formed partnerships. The bacteria became parts of larger cells. This happened about 2 billion years ago. Before that, all life was bacterial, simple, single-celled. After that, complex life became possible. Multicellular organisms, plants, animals, fungi, all built from cells containing former bacteria. So, bacteria are not just ancient, they are ancestral. Every living thing descended from them. Your cells contain their descendants. Your existence depends on bacterial partnerships formed billions of years ago. Bacteria also created soil. Early Earth was rock. Bacteria colonized the rock, broke it down, released minerals, mixed with organic matter from dead bacteria, slowly created soil, made land habitable for plants. They fixed nitrogen, took nitrogen from air, converted it to forms plants can use. Without nitrogen-fixing bacteria, plants could not grow. The entire land ecosystem depends on this bacterial ability. They created and maintain biogeochemical cycles, carbon cycle, nitrogen cycle, sulfur cycle, all driven partly or entirely by bacteria. Remove bacteria and these cycles stop. The planet's chemistry changes. Life as we know it becomes impossible. Bacteria are not just residents of Earth. They are engineers, architects. They built the modern atmosphere, created soil, established cycles, made the planet habitable for complex life. And they continue this work. Every day, bacteria are cycling nutrients, producing oxygen, breaking down waste, maintaining the conditions that allow everything else to live. Right now, bacteria are doing what they have done for billions of years. Living, reproducing, processing chemicals, shaping the planet. The oldest life, the first life, the most successful life. Still here, still working, still essential. Bacteria shaped Earth's past. They shape its present too. They are invisible architects, building and maintaining ecosystems. When anything dies, bacteria decompose it, break it down. A fallen leaf, a dead animal, plant roots, all become food for bacteria. They release enzymes, digest the organic matter, return nutrients to soil. Without bacteria, dead matter would accumulate, pile up. Nutrients would become locked in corpses, unavailable to living things. The whole ecosystem would grind to a halt. Bacteria prevent this. They are nature's recyclers. They break complex molecules into simple ones. Proteins become amino acids. Amino acids become ammonia. Carbohydrates become simple sugars. Then carbon dioxide. Each step releasing nutrients, making them available again. Nitrogen cycling depends on bacteria. Nitrogen is essential for life. It is in proteins, in DNA, but atmospheric nitrogen is unusable. Plants cannot absorb it directly. Nitrogen-fixing bacteria solve this. They have special enzymes. These enzymes break the triple bond in nitrogen gas, convert nitrogen into ammonia. Plants can use ammonia, build proteins, grow. Some nitrogen fixers live free in soil. Others live in plant roots. Legumes host bacteria in root nodules, small lumps on roots. Full of bacteria, the plant provides sugar. The bacteria provide nitrogen. Partnership. Farmers have used this for centuries. Plant legumes, clover, beans, peas. These plants enrich soil. The bacteria in their roots add nitrogen. Next year's crop grows better. Rotation farming, based on bacterial nitrogen fixation. Other bacteria complete the nitrogen cycle. Convert ammonia to nitrate, nitrate to nitrogen gas, return nitrogen to the atmosphere. The cycle continues, driven by different bacterial species at each step. Carbon cycling involves bacteria too. They break down dead plants, release carbon dioxide. This carbon returns to the atmosphere, gets absorbed by living plants. The cycle continues. In oceans, bacteria are primary producers. Photosynthetic bacteria, tiny, floating, invisible, but incredibly numerous. They produce much of Earth's oxygen, more than rainforests. These microscopic organisms are Earth's lungs. They also drive the ocean carbon cycle, absorb carbon dioxide, fix it into organic molecules. Other organisms eat these bacteria. The carbon moves up food chains, eventually sinks to ocean floor, becomes sediment. This removes carbon from the atmosphere. Bacteria controlling climate through their metabolism. Sulfur cycle. Bacteria oxidize sulfur compounds, reduce them, transform them through various chemical forms. Volcanic sulfur becomes sulfate. Sulfate becomes hydrogen sulfide. Hydrogen sulfide becomes sulfur. Bacteria mediating every step. Phosphorus cycle. Bacteria release phosphorus from rocks, from dead organisms, make it available to plants, essential for DNA, for energy molecules, for cell membranes. Iron cycle. Some bacteria oxidize iron, others reduce it. These bacteria can use iron for energy, change its chemical state, create the conditions for iron deposits, for rust. Bacteria also bind heavy metals, remove toxins from water. Bioremediation uses bacteria deliberately. Oil spills. Bacteria eat the oil. Heavy metal contamination. Bacteria absorb the metals. Nature's cleanup crew, put to work solving human problems. In every ecosystem, bacteria are foundation. They process nutrients, make elements available, drive chemical cycles. Other organisms depend on this work. Remove bacteria and ecosystems collapse. Soil health is bacterial health. Good soil is full of bacteria. They form aggregates, improve structure, hold water, release nutrients. Healthy bacterial communities mean healthy plants. Coral reefs depend on bacteria. They cycle nutrients in reef ecosystems, help corals resist disease, break down waste, maintain water quality. Even in your gut, bacteria are ecosystem engineers. They break down fiber you cannot digest, produce vitamins your body cannot make, train your immune system, protect against pathogens. Your gut is an ecosystem. Bacteria are its foundation. These services are invisible, unnoticed, but essential. Life depends on bacterial chemistry, on their ability to transform molecules, to cycle elements, to decompose and rebuild. Right now, bacteria are working in soil beneath your feet, in water nearby, in your body, processing, transforming, recycling. The invisible workers that keep ecosystems functioning, the architects that build and maintain the living world. Bacteria work in ecosystems worldwide. But the most personal ecosystem is you. Your body hosts trillions of bacteria. The numbers are staggering. You have about 30 trillion human cells, but you host 38 trillion bacterial cells, more bacterial cells than your own. You are more bacteria than human by cell count. These bacteria have mass. Together, they weigh about 200 grams, almost half a pound. An organ you did not know you had. Made entirely of microorganisms. Most live in your gut, your large intestine specifically. Conditions there are perfect for bacteria. Warm, moist, dark, full of food. Bacteria thrive. These gut bacteria do essential work. They digest fiber, plant cell walls you cannot break down. They have enzymes you lack, can access nutrients you cannot. They produce vitamins, vitamin K, some B vitamins. You absorb these, benefit from bacterial metabolism. Without gut bacteria, you would be vitamin deficient. They produce short-chain fatty acids. Your intestinal cells use these for energy. Up to 10% of your calories come from bacterial fermentation. Bacteria feeding you from the inside. They train your immune system, especially early in life. Your immune cells learn to recognize normal bacteria, distinguish them from pathogens. This training prevents autoimmune problems later. They protect against pathogens. Good bacteria occupy space, use resources, leave nothing for invaders. Competition keeps you healthy. Diverse bacterial communities resist infection better than depleted ones. Your skin also hosts bacteria, different species in different areas. Oily skin, dry skin, moist skin. Each has specialized bacteria. Skin bacteria produce antimicrobials, natural antibiotics. They fight pathogenic bacteria trying to colonize. Chemical warfare at microscopic scale. On your surface. Constantly, they break down sweat. This creates body odor, not the sweat itself. Bacterial metabolism of sweat. The smell is bacterial waste products. Different bacteria create different odors. Your mouth contains hundreds of bacterial species. They form biofilms on teeth. Plaque is bacterial. Those bacteria feed on sugars, produce acid. The acid erodes enamel, causes cavities, but mouth bacteria also protect. Produce hydrogen peroxide, kill pathogens, convert nitrates to nitric oxide. This lowers blood pressure, benefits cardiovascular health. Mouth bacteria affecting heart health. Every body part has its bacterial community. Unique, stable, yours alone. Your bacterial signature is as individual as fingerprints. Antibiotics disrupt these communities. Kill bacteria indiscriminately, good and bad. The community takes months to recover, sometimes never fully returns to original state. This can cause problems. Without competition from good bacteria, pathogens can take over. Clostridium difficile infections happen this way. Antibiotics kill normal gut bacteria. C. Diff fills the empty space, causes severe disease. Fecal transplants treat this. Take bacteria from healthy gut. Transfer to infected gut. The healthy bacteria recolonize. Outcompete the pathogen. Cure the disease. Treatment using bacteria. Pure ecology. Diet affects gut bacteria. High fiber feeds beneficial bacteria. They ferment it, produce healthy compounds. Low fiber starves them. Changes community composition. Affects health. Probiotics try to add beneficial bacteria. Yogurt, supplements, fermented foods. These deliver live bacteria. The idea is they colonize, improve gut health. Evidence is mixed. Most probiotic bacteria do not colonize permanently. They pass through, might provide temporary benefits, but do not establish lasting presence. Prebiotics are different. These are foods for gut bacteria. Fiber, resistant starch. Feed the bacteria already there. Help them thrive. More effective than trying to add new bacteria. Your bacterial community develops early. Birth method matters. Vaginal birth exposes babies to mother's bacteria. Cesarean babies get different bacteria. This affects immune development. Breastfeeding matters too. Breast milk contains oligosaccharides. Sugars human babies cannot digest, but baby gut bacteria can. The milk is feeding bacteria, not baby, establishing healthy bacterial community. These early exposures set patterns, affect lifelong health. Bacterial communities in childhood influence adult communities. Early investment in bacterial health pays long-term dividends. Right now, trillions of bacteria live on you, in you. They digest food, make vitamins, protect against disease, train immunity. You are not one organism. You are an ecosystem, a collaboration between human and bacterial cells, working together, dependent on each other, inseparable. Bacteria live together in communities, on your skin, in your gut. But how do they coordinate? They communicate without words, through chemicals. This is called quorum sensing. Quorum means minimum number needed. Sensing means detecting. Bacteria detect when enough of them are present. Then they change behavior. Each bacterium releases signal molecules, constantly, small chemicals that diffuse into the surrounding water. When few bacteria are present, signal concentration is low. When many bacteria are present, concentration is high. Bacteria have receptors, proteins that bind these signal molecules. When signal concentration reaches a threshold, the receptor activates. This changes which genes are turned on. The bacterium alters its behavior. This creates group coordination. Individual bacteria respond to population density. When alone, they behave one way. When crowded, they behave differently. All switching together because all detect the same signal concentration. Vibrio fischeri use this for bioluminescence. These bacteria live in squid-like organs, produce light through chemical reactions. But light production is expensive, wastes energy. When bacteria are few, they do not glow. No point. The light would be too dim to matter. But when bacteria are crowded, quorum sensing triggers light production. All bacteria glow together, create bright light, useful to the squid. The squid provides food and shelter. The bacteria provide light. Partnership coordinated by quorum sensing. Pathogens use quorum sensing for virulence. They do not attack immediately. Wait until population is large. Once enough bacteria accumulate, quorum sensing triggers toxin production. Virulence factors, biofilm formation. Coordinated attack overwhelms host defenses. This is smarter than attacking alone. A single bacterium releasing toxins would be destroyed, but thousands releasing toxins together cause disease. Safety in numbers. Coordination through chemistry. Biofilms depend on quorum sensing. Biofilms are bacterial communities stuck to surfaces. Encased in slime, they produce. Dental plaque is a biofilm. So is the slime in drains. Bacteria in biofilms are different from free-floating bacteria. They produce the slime matrix, stick together, resist antibiotics, tolerate harsh conditions, all coordinated through quorum sensing. The signal tells bacteria they are on a surface, crowded enough to justify biofilm formation. They switch on biofilm genes, start producing matrix, building community structures. Different bacteria use different signals. Each species has its own signal molecule. This lets them coordinate with their own kind. Ignore signals from other species. Species-specific communication. But some bacteria can eavesdrop, detect signals from other species, use this to time their own behavior, or to interfere, produce fake signals, confuse competitors, chemical warfare through signal manipulation. Quorum sensing is simple but effective. No language needed, no complex communication, just chemical concentration. Bacteria measuring their own population density through diffused molecules. This is truly decentralized control. No leader, no hierarchy. Each bacterium makes its own decision based on signal concentration, but all make the same decision. Group behavior emerges from individual responses to chemical gradients. Humans try to disrupt quorum sensing as medical treatment, block the signals, prevent bacteria from coordinating, make pathogens attack when still few, easy to defeat. Biofilms never form. Infections cannot establish. Some molecules naturally block quorum sensing, found in certain plants, garlic, cranberries. These might interfere with bacterial communication, prevent coordination. Another way plants defend against bacterial infection. This is chemical ecology. Species interacting through molecules, signals, inhibitors, information flowing through diffusion, behavior changing based on chemical environment. Quorum sensing shows bacteria are not solitary. They are social, respond to their community, coordinate actions, work together toward common goals, all without brains, without planning, just chemistry creating cooperation. The signals are specific, precise, evolved over millions of years. Each species optimized its signal and receptor, made communication reliable, made group behavior possible. Right now, bacteria around you are sensing their populations, detecting signal concentrations, deciding whether to glow, whether to form biofilms, whether to release toxins, all through simple chemical diffusion, communication at the smallest scale, social behavior in single cells, coordination through elegant chemistry. Bacteria communicate with chemicals. Some also use light, not to communicate, to harvest energy. These bacteria have color. Cyanobacteria are blue-green. The color comes from pigments, chlorophyll, phycocyanin. These molecules absorb light, use the energy for photosynthesis. The bacteria make food from sunlight. Purple bacteria are actually purple, deep violet in dense cultures. They have bacteria chlorophyll, different from plant chlorophyll, absorbs different wavelengths, uses infrared light that plants cannot capture. Green sulfur bacteria are brilliant green. They live in deep water, near hot springs, places where little light reaches. Their pigments are extremely efficient, capture almost every photon that hits them. Holobacteria are red. They live in salt lakes. Their red color comes from carotenoids. These protect against intense sunlight in shallow, salty water. The pigments absorb excess light, prevent damage. Halobacteria also have bacterioorhodopsin, a purple protein. This uses light directly, not for photosynthesis, for pumping protons, creating energy without chemical reactions. Pure light to energy conversion. These pigments are why ponds sometimes look coloured. Green water, red water, purple water, all from bacterial pigments. Blooms of coloured bacteria, millions in each drop, enough to tint the water. Some hot springs are rainbow coloured, different bacteria at different temperatures, each with different pigments. Orange bacteria in hot areas, green bacteria in cooler spots, yellow bacteria at the edges, a gradient of colour showing bacterial preferences. The pigments serve specific purposes. Chlorophyll absorbs red and blue light, reflects green. This is why photosynthetic bacteria and plants appear green. The colour they do not use. Wasted light bouncing away. But purple bacteria use green light. Absorb it with their different chlorophyll. They can live under cyanobacteria, using the light that filters through. Different wavelengths, different niches, layered communities, each harvesting what the others miss. These bacterial pigments were the first photosynthetic molecules. They evolved billions of years ago, before plants existed. Bacteria invented photosynthesis, developed the machinery to capture light, convert it to chemical energy. Plants inherited this. Their chloroplasts came from cyanobacteria, ancient symbiosis. The plant cell absorbed a cyanobacterium, kept it. The bacterium kept photosynthesizing, eventually became an organelle, but it still has bacterial pigments. Bacterial photosynthesis, running inside plant cells. Every oxygen breath you take comes from photosynthesis, most from ocean cyanobacteria. Tiny, invisible, but incredibly numerous. They produce more oxygen than all forests combined. These coloured bacteria make Earth's atmosphere breathable. The pigments also protect. Carotenoids absorb excess light, prevent damage from too much energy. Beta-carotene, the orange in carrots, first evolved in bacteria, now found throughout life, always protecting, always absorbing dangerous light. Some bacteria change colour, respond to light conditions, more pigment in bright light, less in dim light. They adjust, optimize energy capture while preventing damage. Scientists use bacterial pigments, fluorescent proteins from bacteria. These glow under certain light, used as markers in research, tagging cells, tracking molecules. The glowing proteins, all from bacteria originally. The variety of bacterial pigments is stunning. Yellow, orange, red, purple, green, blue, brown. Each colour a different molecule, a different strategy, a different way to interact with light. Some bacteria even sense light without pigments. Rhodopsins, light-sensitive proteins, used for navigation. The bacteria swim toward or away from light. Phototaxis, simple behaviour, complex molecular machinery. Colour in nature often comes from bacteria. Pink flamingos get colour from eating shrimp. Shrimp get it from eating algae. Algae contain carotenoids from bacteria. The colour chain starts microscopic. Coral colour partly comes from bacteria too. Symbiotic bacteria in coral tissue. Their pigments add to coral's appearance. Some of the brilliant reef colours, ultimately bacterial. Right now, coloured bacteria photosynthesize. In oceans, in lakes, in puddles. Their pigments absorbing light, converting energy, releasing oxygen. The smallest organisms creating the atmosphere, making Earth habitable, all through coloured molecules. Light captured by bacteria, transformed into life. Coloured bacteria harvest light. But what happens when conditions turn bad? When food runs out? When water disappears? Some bacteria sleep. They form spores. not the same as fungal spores or plant spores. Bacterial spores are survival capsules. The bacterium transforms itself, becomes nearly indestructible. The process is called sporulation. It takes hours. The bacterium stops dividing, copies its DNA, packages one copy with minimal cytoplasm, builds walls around it, layer after layer, creates a fortress. The spore has no metabolism, no growth, no repair. It is dormant, barely alive, more like a seed than an active cell, waiting for better conditions. The walls are thick, multiple layers, peptidoglycan, proteins, a cortex layer, all protecting the DNA inside. These walls resist everything: heat, cold, radiation, chemicals, desiccation. Spores can survive boiling, 100 degrees Celsius. Active bacteria die. Spores survive. Some withstand even higher temperatures. Autoclaves at 120 degrees for 15 minutes. Designed to kill everything. Some spores survive even this. Spores resist radiation. Doses that would shred normal DNA. The spore's DNA is protected, dehydrated, stable, wrapped in proteins that prevent damage. The spore can repair damage once it germinates. Spores survive desiccation, complete drying, no water. Active bacteria die without water. Spores can lie dry for years, decades, possibly centuries. When water returns, they germinate. Spores survive chemicals, alcohol, bleach, disinfectants. The thick walls keep chemicals out, protect the DNA. This is why medical equipment gets autoclaved. Chemical disinfection is not enough. Heat kills spores. The most famous spore former is bacillus. These bacteria live in soil. When soil dries, they sporulate. The spores wait for rain. When moisture returns, spores detected, germinate, become active bacteria again. Clostridium also forms spores. These bacteria live in anaerobic environments: soil, intestines, wounds. Their spores are tough, resistant, can survive in aerobic environments the bacteria cannot tolerate. Clostridium tetani causes tetanus. Its spores live in soil everywhere. Enter wounds, germinate in deep tissue where oxygen is scarce, produce toxin, lock jaw, before vaccines and antibiotics, often fatal. Clostridium botulinum causes botulism. Its spores survive in canned food, germinate in anaerobic cans, produce the most potent toxin known. Tiny amounts cause paralysis. Food safety protocols designed around these spores. But most spore-forming bacteria are harmless. They just use spores for survival, not for causing disease. The strategy is ancient, effective. Wait out bad times in protected state. Some spores have been revived after thousands of years, found in ancient ice, in amber, in sealed containers. When given water and nutrients, they germinate, return to active life, as if no time passed. The oldest confirmed revival is from spores 250 million years old, found in salt crystals. When provided favorable conditions, some germinated, became active bacteria, descended from organisms older than dinosaurs. This longevity is remarkable. Spores are time capsules, bacteria from the past, waiting, preserved by their own biology, ready to resume life when conditions improve. Not all bacteria can sporulate. Most cannot. They must stay active or die. Spore formers have this advantage, can survive catastrophes, wait them out, emerge when safe. Germination is quick. The spore absorbs water, swells. The protective layers break apart. The DNA becomes active again. Ribosomes start working. Within hours, the spore becomes an active bacterium, ready to grow, divide, resume life. Environmental signals trigger germination. Nutrients, warmth, moisture. The spore senses these, determines conditions are favourable, begins the irreversible process of waking. Once germination starts, the spore is vulnerable. If conditions worsen, the bacterium dies, cannot revert to spore form quickly enough. This is the gamble. When to germinate. Too early is dangerous. Too late misses opportunities. Right now, spores wait in soil, in dust, on surfaces, dormant, patient. Some might be decades old, some older, all waiting for the right moment, for water, for nutrients, for conditions that say now, wake now, live again, the sleeping bacteria ready to resume when the world allows. Some bacterial spores cause disease, others help us, especially in food. Bacteria create foods we love. Yogurt is bacterial, milk fermented by lactobacillus and streptococcus. These bacteria eat lactose, milk sugar, convert it to lactic acid. The acid thickens milk, changes texture, creates yogurt. Different bacterial strains create different flavors, different textures. Yogurt makers select specific bacteria, control fermentation, create consistent products. Cheese is bacterial too. Multiple species working together. Some acidify milk, others develop flavor, others create texture. Blue cheese has mold, but bacteria too, all contributing. Hard cheeses age for months, years. Bacteria work slowly, breaking down proteins, fats, creating complex flavors, the sharp taste of aged cheddar, the tang of parmesan, all bacterial metabolism. Sauerkraut is cabbage fermented by lactobacillus, the bacteria naturally on cabbage leaves. Add salt, the bacteria ferment, produce acid, preserve the cabbage, create sour flavor. Kimchi works similarly. Korean fermented vegetables. Multiple bacterial species, creating complex flavors, spicy, sour, umami, all from bacterial fermentation. Sourdough bread uses bacteria and yeast together. Lactobacillus makes the sour flavor. Yeast makes the bread rise. They live in the starter, a culture passed down through generations. Some starters are over 100 years old. Same bacteria, same yeast, continuously maintained. Pickles can be fermented. Not all are. Some use vinegar, but traditional pickles are bacterial. Lactobacillus ferments cucumbers, creates acid, preserves them, develops flavor. Fermentation was preservation. Before refrigeration, bacteria converted food into more stable forms. Milk spoils in days, cheese lasts months, cabbage wilts, sauerkraut keeps all winter. The bacteria make the food acidic. Low pH. This prevents other bacteria from growing. The fermenting bacteria protect the food through their own metabolism. Competition through acid production. Fermented foods are more digestible. Bacteria break down complex molecules, pre-digest the food. Lactose in milk is hard for many adults to digest. Yogurt has less lactose. The bacteria already fermented it. Phytic acid in grains blocks mineral absorption. Sourdough fermentation breaks down phytic acid, makes minerals available. Bread more nutritious. Fermented foods contain probiotics. Live bacteria. When you eat them, bacteria enter your gut. Some might colonize, most pass through, but they might benefit you temporarily. Different cultures have fermented foods. Japanese miso, Indonesian tempeh, Ethiopian injera, all bacterial, all using fermentation, universal human practice, using bacteria for preservation and flavor. Bacteria also make coffee, not the plant, but the flavor develops partly through bacterial fermentation. Coffee cherries get fermented before beans are extracted. The fermentation affects final taste. Chocolate too. Cacao beans ferment in their pods. Bacteria and yeast work together. This fermentation is essential. Without it, Chocolate tastes wrong, bitter, flat. Vanilla beans are fermented. The characteristic vanilla flavor develops during bacterial processing. Fresh beans have little flavor. Fermentation creates the compounds we recognize. These food bacteria are selected, cultivated, passed down. Grandmother's starter, master cheesemaker's culture. These bacterial lineages matter. They create specific flavors, specific qualities. Some bacteria make antibiotics. Nisin from Lactococcus. This kills other bacteria. Food industry uses nisin as preservative. Natural, from bacteria, protecting food through bacterial chemistry. Bacteria also make vitamins, vitamin K, B vitamins. Some fermented foods are enriched by bacterial vitamin production. Another benefit beyond preservation. The fermentation itself creates new compounds, flavors that did not exist in original ingredients, chemical transformations, creating complexity, creating deliciousness. Humans have been using bacteria this way for thousands of years, without understanding what bacteria were, just knowing fermentation worked, knowing it preserved, knowing it tasted good. Now we understand the microbiology, can control it better, select specific strains, optimize conditions, but the basic process is ancient. Bacterial fermentation, creating food, creating flavor. Right now, bacteria ferment somewhere. In a yogurt factory, in a cheese cave, in a crock of kimchi in someone's kitchen. Working as they have for millennia, transforming ingredients, creating foods humans love. Helpful bacteria, making delicious things through simple metabolism. Helpful bacteria make food, but your body must defend against harmful ones. Your immune system constantly watches for bacteria. Your skin is the first barrier. Bacteria cannot penetrate healthy skin. The outer layer is dead cells, thick, waterproof. Bacteria stay on the surface, cannot reach living tissue. But skin also hosts resident bacteria. These good bacteria help defend. They occupy space, use nutrients, leave nothing for invaders. Competition keeps pathogens out. Resident bacteria produce antimicrobials, chemicals that kill other bacteria. They protect their territory by poisoning it for competitors. You benefit from this bacterial warfare. Mucous membranes are different. These line your mouth, nose, lungs, digestive tract. They are living cells, vulnerable, but they produce mucus. This traps bacteria, prevents attachment. Mucus flows constantly. Bacteria get caught, carried away, swallowed, destroyed by stomach acid, or coughed out, sneezed out. The flowing mucus is a conveyor belt removing bacteria. If bacteria get past these barriers, immune cells respond. White blood cells. These patrol your blood, your tissues, searching for bacteria. Neutrophils arrive first. They eat bacteria, engulf them, digest them in internal pouches. Each neutrophil can consume dozens of bacteria before dying. Macrophages are larger, eat more bacteria, also present antigens, bacterial fragments, display them on their surface. This alerts other immune cells, starts adaptive immunity. Your immune system remembers bacteria. After infection, memory cells persist. If the same bacterium returns, response is faster, stronger. This is why you rarely get the same infection twice. Vaccines use this memory. Introduce dead or weakened bacteria. Your immune system responds, develops memory. When real bacteria appear, you are ready, can fight them off immediately. Antibodies target bacteria. These proteins bind to bacterial surfaces. Mark them for destruction. Help neutrophils find them. Block bacterial toxins. Prevent attachment to cells. Complement proteins attack bacterial membranes. Punch holes, make them leak. Bacteria die from loss of membrane integrity. Your blood contains dozens of complement proteins, all coordinating to destroy bacteria. Fever helps fight infection. Higher temperature speeds immune responses, make some bacteria less viable. This is why your body raises temperature during infection. It is helping. Inflammation brings immune cells to infected areas. Blood vessels dilate, become leaky, allow neutrophils and antibodies to enter tissue. The swelling and redness are signs of immune activity, not damage. Defense. Sometimes immune response is too strong. Sepsis. The immune system overreacts, damages your own tissues, trying to kill bacteria but causing harm. This is dangerous, more dangerous than the infection itself sometimes. Your gut is special, full of bacteria, trillions, but these are good bacteria. Your immune system must tolerate them, not attack, while still defending against pathogens. This requires precise discrimination. Your immune system learns which bacteria belong, which are foreign. Regulatory cells suppress inflammation against residents. Allow them to live peacefully. This tolerance develops early. Bacterial exposure in infancy trains the immune system, teaches it what is normal, what is safe. Children raised too clean sometimes develop allergies, asthma. Their immune systems did not learn tolerance properly. The hygiene hypothesis suggests some bacterial exposure is good, necessary even, for proper immune development, not infections. Just normal environmental bacteria, dirt, animals, diverse microbial exposure. Probiotics try to strengthen this defense. Add beneficial bacteria. These compete with pathogens, stimulate immune responses, potentially prevent infections. Antibiotics disrupt bacterial defenses, kill resident bacteria along with pathogens. This can make you vulnerable. Secondary infections happen when good bacteria are depleted. Opportunistic pathogens take over. Your immune system and bacteria are in constant interaction, sometimes fighting, sometimes cooperating, always communicating through chemical signals. Complex relationship, not simple good versus bad. Right now, your immune system patrols, checking bacteria on your skin, in your gut, in your airways, distinguishing friend from foe, destroying invaders, tolerating residents, the eternal vigilance, the constant defense, protecting you from the microbial world while allowing beneficial bacteria to help. Your immune system sees bacteria, but how do scientists study them? How do we count the invisible? The traditional method is culture plates, agar in petri dishes, a gel made from seaweed, nutrients mixed in. This provides surface for bacteria to grow. Take a sample, water, soil, a swab from skin, spread it on the plate, incubate overnight. Each bacterium divides, becomes 2, 4, 8, eventually forms a visible colony, a dot on the plate. Each colony started from one cell. Count the colonies, count the bacteria. Simple, effective, used for over 100 years. But this method has problems. Most bacteria cannot be cultured. They need specific conditions. Nutrients we do not know, temperature ranges difficult to maintain, partners they live with. Without these, they do not grow. Estimates suggest 99% of bacteria cannot be cultured. We can see them under microscopes, know they exist, but cannot grow them in labs, cannot study them using traditional methods. This is the Great Plate anomaly. The difference between bacteria counted under microscopes versus bacteria that form colonies. The numbers do not match. Microscopy shows thousands. Plates show tens. DNA sequencing solved this partly. Extract DNA from samples, amplify bacterial genes, sequence them. This reveals which bacteria are present without culturing them. The 16S ribosomal RNA gene is the standard marker. All bacteria have it, but the sequence varies between species. Like a bacterial barcode, sequence it and identify the bacterium. This revealed incredible diversity. Thousands of bacterial species in one soil sample, hundreds in one gut sample. Most unknown, never cultured. Just DNA sequences proving they exist. Metagenomics goes further. Sequence all DNA in a sample, not just the marker gene. This reveals what bacteria can do, what genes they carry, what metabolism they use. Understanding bacteria through their genomes. This created databases, millions of bacterial sequences, from every environment. Scientists compare new sequences to databases, identify bacteria, build phylogenetic trees, understand relationships. Some bacteria are completely uncultured, known only from DNA, never seen doing anything, just genetic signatures in samples. Candidate phyla, hypothetical organisms, waiting for someone to culture them, or at least understand them better. Fluorescent markers help count live bacteria. DNA stains that only bind living cells, or antibodies that target specific species. View under fluorescent microscope. Count glowing cells, more accurate than traditional staining. Flow cytometry counts millions of cells quickly. Pushes liquid through narrow channel, one cell at a time. Laser detects each cell, sorts them, counts them, analyzes them. High throughput, fast, precise. Single cell genomics is newest. Isolate individual bacteria, sequence each one separately. This reveals diversity within populations, shows some cells have different genes than others, captures variation. Culture plates miss. Microscopy improved too. Confocal microscopy, electron microscopy, atomic force microscopy, each showing bacteria at different scales, different detail levels, revealing structures too small for traditional light microscopes. Live imaging shows bacteria in action. Watch them divide, watch them swim, watch them form biofilms. Time-lapse photography of bacterial life, hours compressed to seconds. The invisible becoming visible. Stable isotope probing tracks metabolism. Feed bacteria isotopes. Heavy carbon, heavy nitrogen, then extract molecules. See which bacteria incorporated isotopes. Know who ate what, who did which metabolic process. All these tools reveal bacterial diversity, complexity, abundance, but also show how much remains unknown, how many bacteria we still cannot culture, cannot study properly. The uncultured majority represents mystery, untapped potential, unknown metabolism, undiscovered antibiotics perhaps, novel enzymes, bacteria doing chemistry we have not imagined. Scientists work to culture the unculturable, try new media, new conditions, co-culture with partners. Sometimes it works. A bacterium grows for the first time. New information, new understanding. But many remain resistant, refuse to grow, stay mysteries. Just sequences, just microscope images. Ghosts in the data, present but unreachable. Right now, scientists are counting bacteria, sequencing them, imaging them, trying to understand the microbial world, the diversity, the complexity, the sheer number of species. Still discovering, still learning, the smallest organisms holding the largest mysteries. Scientists study bacteria, count them, sequence them. But what can we take from this knowledge? What do these smallest organisms teach us? First, that life is everywhere, truly everywhere. No environment too harsh, too remote, too extreme. Where liquid water exists, bacteria exist. They have colonized every possible niche, every surface, every drop of water. This is humbling. We think of Earth as ours, but by numbers, it belongs to bacteria. They outnumber us trillions to one, outweigh us collectively, have been here billions of years longer, will likely be here billions of years after we are gone. Second, that complexity is optional. Bacteria prove life can be simple, minimal, just DNA, ribosomes, membrane. This is enough. Everything beyond is elaboration, specialization, not necessity. We build complexity, cities, technology, culture, all amazing, all impressive. But bacteria remind us that simple works too, that elegance can be found in minimalism, that less is sometimes more. Third, that cooperation happens at all scales. Bacteria communicate, coordinate, work together, quorum sensing, biofilms, symbiosis. They are not isolated, they are social. Communities matter even for single cells. This mirrors human life. We cooperate, build societies, help each other. The pattern exists from the smallest to the largest. Cooperation as fundamental life strategy. Fourth, that we are ecosystems, not individuals. You contain multitudes, trillions of bacterial cells, hundreds of species. They live on you, in you, are part of you. Your health depends on their health. You are interconnected, inseparable from your microscopic residence. This changes identity. You are not just human cells. You are a walking ecosystem, a host, a partner, living in constant collaboration with bacteria. They shape your biology. You shape theirs. Mutual existence. Fifth, that invisibility does not mean insignificance. You cannot see bacteria without tools, yet they run the planet, produce oxygen, cycle nutrients, decompose waste, create soil. Everything depends on their invisible work. This applies to many things. Important processes are often invisible, happening beyond our perception, but real, essential, the unseen mattering as much as the seen. Sixth, that time operates differently at different scales. Bacteria divide every 20 minutes, live fast, evolve quickly, while you age slowly, gradually. Different temporal experiences, different time scales, all valid, all real. Your lifetime contains millions of bacterial generations. They experience rapid turnover, constant change. You experience stability, continuity. Both perspectives exist simultaneously. Different windows onto reality. Finally, that life is chemistry. Bacteria are not mystical. They are molecules interacting. DNA encoding information, proteins doing work, membranes separating inside from outside, chemistry following physical laws, and yet alive, conscious of their environment, responding, adapting. This shows life as emergent, arising from chemistry, not separate from physics, not supernatural, just complexity, organized just right, matter arranged to maintain itself, to reproduce, to persist. There is beauty in this, in the simple fact of bacterial existence. Cells so small yet so complete, so capable, so successful. They are here, now, always, surrounding you, supporting you, sharing this planet. Right now, bacteria cover your skin, line your intestines, float in air around you. They are breaking down dead leaves somewhere, producing oxygen in ocean water, fixing nitrogen in soil. All of it happening constantly, quietly, without fanfare. They ask nothing, expect nothing, just exist. Do their chemistry, divide when resources allow, wait when they do not. Simple existence, pure being. And in their simplicity, there is something peaceful. Life does not require complexity to work, does not require awareness or planning, just molecules, following rules, creating patterns, maintaining themselves against entropy. You are part of this. Your cells descended from bacteria. Your existence depends on bacterial work. You breathe oxygen they made. You digest food with their help. You are connected to them through biology, through history, through the shared fact of being alive. Sleep tonight, knowing you are not alone. Billions of bacteria share your journey, living on you, helping you, part of you, the smallest organisms, essential, ancient, ever-present, simple life, everywhere.